So again, this is a reminder कि cell वो chapter असा पर. This is the basic fundamental thing that we require for all the chapters that we will be learning next. Okay, animal classification, structural सुद्धा organization in animals पहिले पय आम्ही त्या सगळ्यांक खातीर this is the base. This is the place from where we are starting with everything that is required to understand everything in depth. Okay, even plant physiology असा पय then photosynthesis जो part असा तितून सुद्धा जे गोष्टी important असा that is there here. Okay, so please pay attention to everything because हेंच्याकून deep किती म्हणतात ते information we will be learning in the other chapters 11th of course थोडे गोष्टी 12th in depth असा so you should know these things. Okay, I have already told you molecular biology जेन्ना हांव माझ्या फर्स्ट बॅचाक शिकयताले सो तांकां म्हणजे 12th एक चॅप्टर आसा मॉलिक्युलर बायोलॉजी सो तांकां जेन्ना हांव शिकयताले फर्स्ट टाइम 12th बायो सो दॅट टाइम इट लिटरली टूक मी 1 फुल मंथ आणि एक फुल मंथ म्हणजे कितले लेक्चर्स जातले 3 कितले well almost 12 lectures. ओके कित्याक सांग तेंची बेसिक्स 11th क्लियर नासली म्हणून ओके दॅट इज द रिजन वाय वी आर पुटिंग एक्स्ट्रा एफर्ट्स इनटू एवरीथिंग ओके सो प्लीज मेक शुअर की हे तुमकां सगळे तकलेन वता सो दॅट फुडले चॅप्टर सुद्धा तुमकां सारखे कळटले ओके नॉट ओनली ऑफ 11th स्टँडर्ड बट ऑल्सो ऑफ 12th स्टँडर्ड येस fine so let's move ahead with this cell chapter and today we are going to learn about cell wall. Okay, cell wall is present in plants and this is the outer covering which is acting like a skeleton, right? If cell walls are nashillo ni जाल्यार okay imagine there is a animal cell and there is a plant cell आणि दोन्ही आम्ही किती करता उदकान घालता okay उदकान घालतोय किती जाता since water concentration is less inside the cell water will start going into the cell right हळू भितर वचपाक लागतलो but animal cell after some time will burst. Okay, so कितलो फुट्टलो but plant cell will not burst. This is because of presence of cell wall. Okay, cell wall is giving it its rigidity and the reason why the plant cell will never ever burst in any solutions. Is this clear? हा आता खूप किती difficult खंयचे तरी hard acid बीन हाडून घातले नी concentrated then it will burst. Okay, सामके concentrated पण तितून आता full cellus मागीर damage जातलो. Then you can't use that cell for any other purpose. Okay, is this clear? Fine. So what we were discussing here, definition, non-living rigid structure forms an outer covering for the plasma membrane of fungi and plants. Okay, then role obviously it gives shape and it is protecting, okay, protecting the cell from the mechanical damage and infections. Okay, then you know right a plant cannot move from one place to another so obviously infections जावपाक शकता. For example, if you sit on a place which is very dirty, then you might get some skin allergies, right? If you touch some fungus, again you might get some respiratory allergy or maybe even touch केला तर skin allergy जाऊ शकता. So आम्ही किती करता आम्ही avoid करता. So we are moving away from that particular threat. पण plants आणि that is not possible. Okay, they can't move, they can't run. म्हणून they have this inbuilt protection for them. Okay, that is cell wall. Fine. So this cell wall they are also helping in cell to cell interaction and it is providing barrier to undesirable macromolecules. Macro म्हणजे किती big, right? Macro means big, micro means small. So जे किती macro मोठे molecules असा which are not required into the cell त्यांका ते आणि entertain करना it is not allowing it to pass inside. Okay, is this clear? Role is clear to everybody? Okay, so definition झाले, role झाले. Next is about the chemical composition. Next is chemical composition. So हांगा दोन हेंचे chemical composition दिला, एक algae and second they have mentioned about the plants. Okay, so algae खंयचे chemicals असता in the cell wall? Cellulose, galactans, mannans and minerals like calcium carbonate. These all are present in cell wall of algae. Fill in the blanks येऊ शकता. Which of the following is present in the cell wall of algae? Okay. So तीन खंयचे तरी answer correct असतले, एक खंयचे तरी wrong असतले. Okay, not present or is present. So you should read the question very carefully. Okay, and accordingly you should answer. So next is about other plants. Other plants म्हणजे सगळे तुमचे मोनोकॉट, डायकॉट. तिथून किती असतात? तिथून सेल वॉल असता cellulose, hemicellulose, pectins and proteins. Okay, what are the common things here? Cellulose is common. Then other things, proteins ना बाकीचे सगळे is not so common. Okay, what do you have? Hemicellulose. आणि one more thing, you can say hemicellulose is the key character of plants. Okay, सगळ्या प्लांट्सांनी हेमिसेल्युलोज हे असतात. So if you see hemicellulose, you should blindly say that it is for the plants and not for the algae. Algae किती असता? Algae असता cellulose, galactans, mannans, minerals like calcium carbonate. Okay, is this clear? किती doubt असा हांगा? What is? They are just names of the chemicals which are present. And kind. These all are all chemicals. And kind. See, their structure is little bit different. Cellulose and hemicellulose is a barik so structure. So difference. So that chemistry part we are not focused. Okay, that is not my subject. Okay, so just remember these are the chemicals which are present inside the cell wall. Okay, then structure. Ata ho cell wall is little bit different when the plant is very young. It is little bit different when the plant is turning old, right? जसे जसे plant वळले जावपा लागता, of course it is becoming more and more rigid, right? एक वळले, can you go and just pluck one coconut tree and come home? No, okay, but can you go and pluck one grass and come home? Yes, that is easy, right? So अशाच आम्ही म्हणू शकता that किती ते structure of cell wall is different depending on what type of plant it is and also it is at which stage. बारीकशे असं आता mango plant, if it's very small आत्ताच लायला, then you can pluck it, right, anytime. But when it becomes very big, that time you cannot do anything that particular plant, right? So this is what I'm trying to highlight here. In a young plant, what happens? Primary wall. Okay, primary wall. आता cell wall ज्यांना young plants नसतात त्याका आम्ही किती म्हणता? Primary wall. It is capable of growth. Okay, हरशी cell wall grow जायना. Cell wall जितली असा पण तितलीच उरता. But when this plant is very young, त्यांना किती जाता? It is capable of growing. Okay, then mature plant do not show growth. Secondary wall is formed on the inner towards membrane side of the cell. So ही inner side असा पळय, थंय किती जावपा लागता? Secondary wall जावपा लागता. For example, पहिली the cell wall is very thin. Okay, so ते त्यांना grow जावपाक शकता. मागीर किती जाता? भितरल्या वाटेन somewhere inside. So we can say this is the cell wall in the earlier stage. Okay, मागीर किती जावपा लागता? Now it is growing. Okay, grows down. It is now becoming of this size, right? आता किती जावपा जाय तेका? आता तेका घट जावपा जाय. So what is happening? Secondary wall is forming inside, and now it is becoming more and more rigid. Okay, inside. हांगा भायल्या कितून जायना ते भितर किती जावपा लागता? हळूहळू they are going to become thick. Okay, is this clear? Then middle lamellae. So there is one more thing or one more statement which you have to remember. One more word, keyword you have to remember is middle lamellae. Can you see this? Middle lamellae. This is one cell. This is another cell. आणि असे खूप cells असतात पय लागी. Okay, so this line between these two cells is called middle lamellae. It is called as middle lamellae and it is made up of calcium pectide. Made up of what? Calcium pectide. So what is the use of this middle lamellae? It is acting like a glue. Cell एकामेकाच्या पयस वचपाक जायना न्ही, they should be glued together. So what is the work of this middle lamellae? It glues the different neighboring cells together. Okay, so that is the use of middle lamellae. Now there is one more term which you have to remember, that is plasmodesmata. Okay, can you see these openings here? Okay, so a small opening which is there between the cells, that is actually plasmodesmata. This is like a khidki. Okay, खिड़की खबर असा नाही खिड़की window. Okay, this is like a window, so whatever the neighbor wants they can exchange with each other. Okay, so this plasmodesmata is very very important, and this connect the cytoplasm of the neighboring cells. So whatever that is there in this cytoplasm, whatever that is there in this cytoplasm, जर एकामेकाक जर त्याची गरज असा नी any molecule, then they can exchange via this plasmodesmata. Okay, is there any difficulty here? Yes, clear. So do one thing, please draw this diagram, because what diagram have notes on घालो ना. Notes you got your notes? No. Maybe by tomorrow we'll get it. Okay. So cell, you should draw cell, plasma membrane, cell wall, and then between them you can draw middle lamellae, and also you have to draw plasmodesmata. Yeah. Is this clear now? So next thing what we are learning about is endomembrane system. Okay. If you are not feeling well, you can just lie down. No problem. You okay? No, I feel that you're not well today. That's all. You can lie down. No problem. Okay. Okay. So what we have next is endomembrane system. आता endomembrane system म्हणजे किती? So they all are the organelles that work together like a team. Okay, their functions are dependent on each other. So which are the cell organelles which are coming into this category called as endomembrane system? Number one, can you read it? It is endoplasmic reticulum. Second, Golgi apparatus. Third, lysosome. Fourth is vacuole. म्हणजे किती सांगू शकता तुम्ही? Whatever role that is performed by this ER, it is actually dependent, it is actually carried forward by this Golgi complex. And जे Golgi complex करता, that is again carried forward by lysosome. Lysosome किती काम करता? Carried forward by vacuole. Okay. For example, I am preparing some food. Okay, then I'm parceling it. Swiggy yeta, so that is the another organelle, Swiggy or Zomato or whatever. So they are delivering that to someone else. मागीर ते payment करता कोण? GPay वयल्यान. So we all are working together. Okay, that app, Swiggy app, and also me. Okay, so this is like how everything runs also here in this endomembrane system. Okay, all these organelles are working together. Fine. So mitochondria, chloroplast, peroxisome, here organelles अशी आसा which is not coming in this category. So their functions are independent from all these endomembrane systems. Is it clear to everybody? किती doubt आसा हांगा? Yes? Clear? Okay. So the first thing which we are learning now is endoplasmic reticulum. Okay, it is endoplasmic reticulum. Do you want some short break or something of 5 minutes? It's okay, you can tell me. खाए ना तुमका. Do you want a break? You? At 4 o'clock. Okay, at 4 o'clock I'll give you breakdown. So now we have 15 minutes. So for 15 minutes, please concentrate and then we can take a break. Why I'm telling concentrate, concentrate? Because it's easy गोष्टी असा, but you are not going to go home and learn this, right? कोण घरा वचून स्टडी करपाचो आसा जर हे कोण आयकना जाल्यार आसा कोण ना न्ही सो दॅट इज द ओनली रिजन वाय आय वॉन्ट यू टू बी अटेंटिव्ह इतलेच तुम्ही हांगा येयल्या तुम्ही फी पे केल्या यू गायज आर लिटरली इन्वेस्टिंग युअर टाइम हियर सो ऑब्वियसली तेच्या खातीर तुमकां अटेन्शन मातशें तरी पे करचें पडटा ओके एंड इट इज फॉर युअर गुड तुमकां मार्क्स पडले एवरीबडी वील प्रेज यू नो बडी वील से हेची बायो टीचर खूब बरी आशिल्ली म्हणून हेका फुल मार्क्स पडले नो बडी इज गोइंग टू से दॅट सगळी म्हणटली की तुम्ही हार्डवर्क केले तुम्ही स्टडी केले म्हणून तुमकां मार्क्स पडले ओके नो बडी इज गोइंग टू गिव मी द क्रेडिट okay but I will feel very happy कि हाँ चलो my students are scoring good marks okay fine so please only for 15 minutes then we'll take a short break yes so endoplasmic reticulum or ER this looks like a tiny tube like or tubular structure okay त्याचे structure किती असा tube like or tubular structure and it is just present here scattered here and there in the cytoplasm आतापर्यंत सगळ्यांना खबर असतली किती right then so ER is divided into luminal and extraluminal parts so ER किती असा it is divided into two parts luminal आणि extraluminal luminal म्हणजे किती inside ER that means whatever that is there inside the ER that will be luminal okay and extraluminal म्हणजे किती whatever that is present outside here in the cytoplasm that is extra in bio means outside okay in biology extra means outside so हांगा कितें म्हणटा आमी extraluminal म्हणजे outside the luminal area yes is this clear that is cytoplasm okay now RER SER you all know the full form that is rough endoplasmic reticulum आणि smooth endoplasmic reticulum. So these are two types of endoplasmic reticulum present based on whether they have ribosomes or not. Okay, if the ribosomes are present, they are called as rough endoplasmic reticulum. Can you see this diagram? In this diagram, these some folds are having this small white or yellow color small vesicles, right? तुमका सांगायला नाही last time vesicles म्हणजे any structure that is circular. Okay, any structure that is round in shape, circular in shape, त्याका आम्ही किती म्हणता vesicle म्हणता. सांगा तुमका बारीक बारीक yellow vesicles दिसता. So they are what? They are nothing but ribosomes. So ER चं असलं part जिथून हे ribosome attach असा त्याका आम्ही म्हणता rough endoplasmic reticulum. आणि जर if these ribosomes are not attached, then they are called as smooth endoplasmic reticulum. Okay, so ribosome attached to their outer surface, that is RER. RER is frequently observed in the cell actively involved in protein synthesis and secretion. So what is the role of RER, rough endoplasmic reticulum? Protein synthesis and secretion. Protein they synthesize करता, तयारी करता, and also they are secreting it out. Okay, again, they are extensive and continuous with the outer membrane of the nucleus. So where they are present, this is the nucleus, right? And this is the nuclear membrane. So they are continuously present outside the nuclear membrane. So first point किती सांगता? First point सांगता is structure की त्याचे कोण present असा ribosome. Second point is telling us about the function of RER. The last one, it is telling us where it is situated or which is the area where this RER is present. Okay, RER concept clear zalo? Yes, okay. Now SER. SER, here there are no ribosomes. That's why they appear very smooth in texture. Okay, clear na tha? Ribosomes na, and that's why they are appearing very smooth. Major site for synthesis of lipids. In RER, किती synthesize जाता? Protein synthesize जाता आणि protein secrete सुद्धा जाता. And in SER, only lipids are getting synthesized. Okay. By the way, these are the differences between RER आणि SER. सगळ्यात पहिले difference किती येतलो? Ribosome present, ribosome absent. Second किती येतले? Yes, synthesis. एक protein synthesis, दूसरा lipid synthesis. Okay, then in animal cell, lipid like steroidal hormones are synthesized in SER. So in some of the animal cells, SER किती करता? Lipid like steroid hormone सुद्धा तो synthesize करू शकता. So it can synthesize what? Lipids, and it can also synthesize lipid like steroidal hormones in the case of animal cell. Is this clear to everyone? किती doubt असा हांगा? No, right? So in short, endoplasmic reticulum किती करता? It is synthesizing some chemicals, right? Proteins or lipids, depending on whether it is SER or RER. Fine? Clear झाले? This diagram is clear to everybody? कोण RER, कोण SER, what is this round structure? ये सगळ्यांना कोण आहे? This is nucleus, by the way. Yes? Okay. Next is about Golgi apparatus. Let me tell you one thing. Only these are the things which are difficult in this chapter. Okay, आणि difficult म्हणजे किती सांग? केन्नाच जर वाचूना, केन्नाच जर वाचून understand करून diagram बीन काढूना, then it is difficult. But if you read it today, okay, if you listen it now, then if you revise it only 5 minutes before starting the next class on Friday, then this is like done. दोन तीन points असा फक्त सगळ्यांच्या under. So सगळे points जर परत परत वाचले, just 5-10 minutes before your bio class, everything will be sorted. Okay. Then next is about Golgi apparatus. So this was observed by whom? Camillo Golgi in 1898. Hence the name. Okay, त्याच्या नावा वयल्यानो, scientistच्या नावा वयल्यानो Golgi apparatus, this name is given. So what he observed? Densely stained reticulate structures near the nucleus. So nucleusकडे एक खूप dark stain reticulate shape तेका किती दिसलो? Structure दिसलो. कोणाक दिसलो? Camillo Golgi in the year 1898. 98. Okay. Then structure of this Golgi apparatus, it is disc shape. Okay, disc shape sacs. Sac खबर आसा न्ही sac. Okay, it's like disc shape sacs or cistern. तेका आमी कितें म्हणटा cistern सुद्धा म्हणटा. Please highlight this cistern. Very important term. Okay, so cistern is a very important term. हे cistern term आणि सुद्धा परत परत पुढे येतलो. Okay, cistern म्हणजे कितें something which looks like a disc and it is present in a sac like form. Okay, असो. Fine, this type of arrangement will be called as cistern. Okay, so don't forget this name. So कितले आसा त्याचे diameter 0.5 micrometer to 1 micrometer. Okay, then these are stacked parallel to each other. So a cistern आसा पय, this is one cistern, second, third, fourth, fifth. So ते कितें parallelly arranged आसा एकामेका बरोबर. Varied number of cisternae are present in the Golgi complex. So depending on which type of cell it is present, in which animal it is present, हे cisternae in number कितले असतले, that is differing. Okay. Then Golgi cisternae are concentrically arranged near the nucleus. So location खंय आसा? They are present near the nucleus. Okay. Then Golgi, very important part. This is the structure of Golgi apparatus. तेका दोन faces असता, एक cis आणि दुसरो trans. Can you see this? This is cis face, this is trans face. So हो जो diagram असा पय तुमच्या textbookan, so you can label this as cis face and label this as the trans face. Okay, this is cis face, cis or forming face, cis or forming phase, and this is trans or maturing phase. Where is your textbook, Benjamin? Okay. Cis or forming phase, trans or maturing phase. Okay. Yes, so why I'm telling you this part? Because of course structure is important to learn about any organelle. Second, हे जे arrangement असा पय, it is present near the nucleus, cis phase असा, trans phase असा. So what is happening here? These all are playing very important role in performing the function. Okay, so what is the role or function of Golgi apparatus? So they are primarily used for packaging material. So तुम्ही जर Golgi apparatusचे role बरयता, and if you are not mentioning about packaging, then you will lose your marks. Okay, the question is for 2 marks. Write function of Golgi apparatus, and जर तर तुम्ही packaging material हे word use करना, you are not going to get full marks. They will give you 1 or 1.5 maximum. Okay, so perform function of packaging material to be delivered to the intracellular target or secreted outside the cell. So outside or inside the cell, खंयूय तेंका जे transport करपा जाय पय, whatever it is, that packaging of that material is done by this Golgi apparatus. Okay, what was the role of endoplasmic reticulum? Very good, secretion, right? Synthesis, secretion. Okay, then so materials to be packed in the form of vesicle. Vesicle म्हणजे किती असतले? Round ball like structure, right? So they will be packed in a, आम्ही कसे बॉक्सान पॅक करता, हांगा त्याचे बॉक्स शेप काय ना, it is packed in a circular vesicle. Okay, from the ER fuse the cis phase of the Golgi. So किती जाता, they are, can you see this cis? Okay, can you see this vesicle here? सो तो कितें जाला तो हांगा पॅक्ड जाला. ओके, पॅक्ड एट विच स्टेज? सिस स्पेस. ओके, सिस स्पेसा कडेन तो कितें जाता पॅक जाता इन द वेसिक्युलर फॉर्म आणि मागीर मूव टुवर्ड्स द मॅच्युरिंग ऑर द ट्रान्स फेज. सो नाव इट इज मूविंग फ्रॉम हियर टू हियर. Okay, so what you can do is you can put a arrow from here to here. Okay, from here to here you can put a arrow and you can say packaging material in a vesicle form, transport of packaging material in vesicle form. Okay, so now we will understand how ER and Golgi are placed very close into the cell. Okay, दोन्ही किती असा एकामेकाच्या खूप लागी असा कोण endoplasmic reticulum आणि Golgi apparatus. So number of proteins which are synthesized by the ribosomes which are present on ER म्हणजे कोण असतलो हो RER. Okay, are modified in cisternae of Golgi apparatus. Okay, so what is produced here? Proteins जे RER produce करता त्याचे किती जाता they are little bit modified inside the cisternae of Golgi apparatus before they are released in the trans phase. So ते pack करून पुढे move करपाक or पुढे transport करच्या पयली किती जाता they are little bit modified. Where they are modified? In the cisternae of Golgi apparatus. So RER is producing that or synthesizing that protein. That protein is transported to the cisternae of the Golgi apparatus. So it is modified and then it is packed in the vesicle form and then it is transported towards the trans phase. Is this clear? Okay, now you know why ER and Golgi are placed very close to each other. Work question most of the time is asked for 2 marks. Please highlight this. Okay, packaging material and under that these 2 points are coming. Okay, many times star you can put a star also. Maximum time so question yetas. Okay, why ER and Golgi are placed close to each other in the cell? Okay, next Golgi apparatus is an important site for the formation of glycoprotein and glycolipid. So that is another function of the Golgi apparatus. So किती जाता है? Formation of glycoproteins and glycolipids. Okay, so next is about lysosome. Before that, we'll take a small 5 minutes break. Okay, we are on slide number 42. We have to reach slide number 62. I just soplala baratle, but जावना झालं रे काय problem. Okay, yes, you can take a break. So which are the organelles we have studied till now? plasma membrane, cell wall, then endoplasmic reticulum, which are two types, rough and smooth, and then last is Golgi apparatus. Okay. So now we are moving towards the next organelle. Lysosomes. Okay. So number one thing which you have to remember is they are called as the suicidal bags. किती म्हणतात ते का? Suicidal bag. This word is not there in your textbook, but please you can write it down with your pencil. Okay, suicidal bags or cell recycling center. Suicidal bags or cell recycling center. Okay. So these are the membrane bound vesicle structure which are formed by the process of packaging in the Golgi apparatus. So where they are formed, they are formed during the packaging process which is done in Golgi apparatus. Okay. The isolated lysosome vesicles have been found to be rich in almost all type of hydrolytic enzymes. So त्याच्या भितर there are presence of some what? Hydrolytic enzyme. Please highlight this hydrolytic enzyme. Hydrolytic enzymes म्हणजे किती आहे? Hydrolysis. Hydrolysis example कोण? Lipases, proteases and carbohydrates. Okay, ASES last that means they are enzymes. They are what? Enzymes, and they are going to digest or cut or eat up the respective molecule. For example, lipases it is going to cut down lipase. Magir protease it is going to cut down proteins. Carbohydrates are going to cut down the carbohydrates. Okay, is this clear? The functions are clear? Yes. Now, so optimally active at the acidic pH. So what will be the condition or what will be the environment inside lysosomes? It will be acidic in condition because there are so many enzymes which are present and they are referred as suicidal bags or cell recycling center. So हे जे enzyme असा they are capable of digesting all these molecules. Okay, वो actually एक बारीक सो हांगा process असा which is explained, but this is extra. Okay, this is extra हे तुमका ना. So have you heard of endocytosis? Food particles ले, okay, food particle भितर घेतले, that process is endocytosis. ते food particle आता broken down into lysosomes आणि जे किती useful असा plant cell खातीर that is used by the cell, and whatever that is not useful that is sent outside the body via elimination process. Okay, this is one of the major function of lysosome. हे mention केले ना हांगा, but this is one of the important thing which we have to know. Okay, so एक काम करू शकता तुम्ही, just one word you can write down near lysosomes, that is help in endocytosis. Help in endocytosis, small dash digestion vacuole, digestive vacuole, small dash digestive vacuole. Okay, next is about the vacuoles. And vacuole हमको खबर सा, they are like the empty space. They are present in plant cell animal cell and they are absent. Okay, so vacuoles are not found in the animal cell, and even if they are present, it will be very less, low hostile, like a small dot. Okay, so vacuole is the membrane bound space found in the cytoplasm. Membrane bound म्हणजे किती by the way त्यांका covering असा म्हणून आम्ही त्यांका किती म्हणता membrane bound म्हणता. Okay, so they are containing what? So vacuolaच्या भितर किती किती present असता water, sap, excretory product and other materials that are not useful to the cell. Okay, cell जे गोष्टींची requirement ना, all those things are kept in the vacuole. So in short, you can say that all the toxins are stored in the vacuole. Okay, all the toxins are stored in the vacuole. Okay, मागे vacuole is bound with a single membrane called tonoplast. First lecture तुमका विचारलेले what is tonoplast? So tonoplast is nothing but the outer structure or the outer membrane of the vacuole. Okay, for example, here diagram. You can see this. This is the vacuole, right? So ह्यो भायलो layer जो असा पय तेका आमी कितें म्हणटा tonoplast. This is clear to everyone? Yes? Okay. So please highlight tonoplast. Okay. So since they are present in plant cell, we are going to discuss about few points about vacuoles in plant cell. So hanga किती दिला vacuoles in plant cell, vacuole in amoeba and vacuole in protist. Okay, these are three one marks question. What is the role of vacuole in plant? What is the role of vacuole in amoeba? What is the role of vacuole in protist? So in plant किती करता almost 90% of the volume is of the vacuole. हे आम्ही diagram already पहिले असा. Then tonoplast जो outer layer असा तो किती करता? It is helping in the transportation of number of ions and other minerals. So tonoplast किती करता? Transportation inside or outside the vacuole, it is facilitated by the tonoplast. Okay, so this is happening against the concentration gradient म्हणजे even it can move from the lower concentration to the higher concentration. Okay, so hence their concentration is significantly higher in the vacuole than in the cytoplasm. So किती होता vacuolan actually जे किती गोष्टी सेलाक necessary नसता पय everything is thrown into the vacuole. So किती होता vacuolan त्यांचे concentration वाढता. So always whenever something which is coming inside the vacuole is always against the concentration gradient. Okay, म्हणजे they are moving from the low concentration to the higher concentration. Okay, is this clear? किती difficulty आहे सांगा? Is there anything that you have not learned? In plants, vacuole clear sa sagna? Yes? Okay. Now about amoeba. So in amoeba, vacuole is helping in the contractile vacuole. So देखा आम्ही किती म्हणता? Contractile vacuole important for osmoregulation and excretion. Okay, it is required for osmoregulation and also in the excretion process which takes place in the amoeba. Next is about protist, and in protist it is helping to engulf the food. Okay, प्रोटिस्टान जे vacuole असतात तेका आम्ही food vacuole म्हणता, and it is formed by engulfing the food particle. Food particle ज्यांना amoeba असं open करून भितर घेता पय त्यांना ते एक food vacuole तयार जाता. Okay, so this is helping in the intake of food in the case of protist, and amoeba it is osmoregulation and excretion, and in plants you can say all the toxins are stored inside the vacuole. Okay, and what is the outermost layer of vacuole? Tonoplast. Is there any difficulty here? Next is about mitochondria. Okay, we'll study about mitochondria, then we'll stop. Any difficulty till now? Okay. Highlight केला tonoplast. And plant, amoeba and protist also you can highlight. And amoeba, contractile vacuole, protist, food vacuole. Yeah, this can come for the match the pairs. Okay, match the pair. Amoeba, contractile vacuole, protist, food vacuole. Okay. Next is about mitochondria. सगळ्यांक खबर आसा mitochondria is the powerhouse of the cell. So hanga we are learning the structure of mitochondria, and this is very important from the point of view of your next unit, that is plant physiology. Okay, so please pay close attention to this organelle. Okay, so mitochondria किती आसा? एका सेलान कितले mitochondria आसतले, that will be changing depending on the physiological activity of the cell. एक mitochondria आसा, 2 आसा, 3 आसा, 4 आसा, 5 आसा, that depends on the physiological process. Okay. Second point किती आसा हांगा? Mitochondria, they are dividing by fission. So they have to divide also. So they divide कशे जाता? By the process of fission. Okay. Magir, what is the role of mitochondria? Obviously, they कितें जाता? Site of aerobic respiration, and in this respiration they are producing something which is very important, जेका आमी कितें म्हणटा? ATP. What is the full form of ATP? Adenosine triphosphate. म्हणजे हांगा कितले phosphate आसतले? 3. Triphosphate. There is also something called as ADP. That is adenosine diphosphate. म्हणजे कितले phosphate असतले? 2. And there is also something called as AMP. म्हणजे हांगा कितले phosphate असतले? Adenosine monophosphate. Means how many phosphates? Only one phosphate. Okay. So अशे चिंतनाका कि there is only ATP and there is nothing else. There is ATP, ADP and also AMP. Okay, depending on how many phosphates are attached to the adenosine. Yes, is this clear? Fine. So किती जाता हांगा ATP produce जाता, and obviously that is the reason why it is called as powerhouse of the cell. Matrix also possesses single circular DNA molecule. किती आसा त्याच्या भितर? Single circular DNA molecule. So DNA is usually present in nucleus. Nucleus is the main region ज्या जाग्यार कितें present असता? DNA present असता. हे जे कितें आसा पळय DNA, these are called as extra chromosomal DNA. तांकां कितें म्हणटा आमी? Extra chromosomal DNA. So chromosomal DNA खंयचे? Which are present in the nucleus. आणि extra chromosomal DNA म्हणजे खंयचे? Which are present in mitochondria. And also there is one more organelle जिथून DNA present असता, that is chloroplast. Okay, these are what? Extra chromosomal. Extra म्हणजे किती सांगलेलं तुमकां हांवें वदोळ? Outside, right? Outside. So chromosomal DNA present in nucleus, extra chromosomal DNA present in mitochondria and chloroplast. Is this clear? Okay. Which of the following organelle there is presence of DNA? Also question येलो. Option 1 nucleus, option 2 chloroplast, option 3 mitochondria, option 4 lysosomes. So खंयच्या हितून DNA ना? This lysosome. Okay, so हे खबर आसपाक जाय सगळ्यांक. The extra chromosomal अशें काय मेन्शन करूना. Then you should understand that they are asking about in which organelle it is not present. And if they mention which of the following there is presence of chromosomal DNA. Option 1 nucleus, option 2 mitochondria, option 3 is chloroplast, option 4 is vacuole. Answer किती? Very good. It is nucleus. Okay. Now next is about structure of mitochondria, and this we will do in next class, or now only 2 slides are remaining. Then last is last last years, this will do next time. Okay, this just these 2 slides we will finish. So ankide asa shape and size, it is variable. Mitochondria एक particular shape आणि particular size नसतलो अशें काय ना, but typically it looks like a sausage shape. कित दिसता ते? It looks like a sausage or cylindrical shape. Diameter is how much? 0.2 ते 1 micrometer. Okay, average किती असू शकता? 0.5 micrometers, and the length is 1 or 4.1 micrometer. Okay. Diameter कितले असा? Overall average 0.5 micrometer. लेंथ असू शकता 1 or 4.1 micrometers. Okay, then they have double membrane. Very important statement. किती असा भायले layer, you can see there is one layer outside and there is one more layer inside. So there is outer membrane and there is inner membrane present. Okay, so किती असा दोन membrane present असा तेका outer and inner membrane. Similarly, there is one more organelle जेका दोन membrane present असा and that is chloroplast. ओके, अगेन मायटोकॉन्ड्रिया आणि क्लोरोप्लास्ट यांच्या दोघांच्या भितर खूपशी सिमिलॅरिटीज असा. ओके, एक तर एक्स्ट्रा क्रोमोझोमल डीएनए प्रेझेंट असा. सेकंडली दे बोथ हॅव डबल कवरिंग और डबल लेयर्स. ओके, डबल मेम्ब्रेन. इज दिस क्लियर? येस. ओके, देन so there is a membrane जेका आम्ही किती म्हणता outer membrane and the inner membrane. So this now is divided into two particular section, outer membrane, outer compartment आणि inner compartment. Okay, so this is the inner membrane, this is the outer membrane जे covering असा आणि जे भितर असा पय that is the inner compartment आणि हांगा मधी असा तेका किती म्हणता आम्ही outer compartment. Okay, so again in detail we will see inner compartment and किती असा this is filled with dense homogeneous substance called matrix. So हेका किती म्हणता आम्ही matrix. Green part दिसता तुमका, that is matrix which is present in the inner compartment. Please highlight this matrix. This is one of the most important term. Okay, मागे क्रिस्टे जे असा which is this formation. Okay, असे coil formation पे क्रिस्टे that is increasing the surface area. किती यूज असा त्याचं surface area increase करता so that त्याचे functioning सारखे conduct जावपाक शकता. Okay, then outer compartment किती असा forms continuous limiting boundary of the organelle. So that is like a covering layer. भायलो जो लेयर असा भायले जे कवरिंग असा तेका आम्ही किती म्हणता outer compartment. आणि जे किती भितर असा in the matrix that is called as the inner compartment. Okay, is this part clear to everybody? Now, two membrane have their own specific enzyme associated with the mitochondria function. So त्यांच्या भितर specific किती म्हणतात ते chemicals or specific enzymes असा and these enzymes are playing some very important role. आणि ह्यांचे जे रोल असा पळय that we will be actually understanding in the plant physiology part. Okay, थंय आम्ही everything in detail about the enzymes we are going to study. पण आतापर्यंत तुमका full structure कळो mitochondria, inner compartment and outer compartment. Inner compartment किती म्हणता आम्ही matrix म्हणता आणि cristae is helping in the increasing the surface area and also there is outer compartment which is nothing but the continuous outer covering. Okay, is this clear? Kiti doubt asa? No. Okay, so we'll stop here for today. Just give me one pencil. What is the date? 12. Benjamin, Vidal, Joseph is absent, Yuzico, Daniela, Lisa, Shania absent, Skyla absent. Okay, check. Thank you. Yeah, bye. Yuzico, just give me the phone. Thanks.